Welcome to Biochemistry Unfolded, From Atoms to Algorithms, an online podcast on the topic of biochemistry and biophysics. Welcome back to Biochemistry Unfolded, From Atoms to Algorithms, the podcast where we zoom in on the fundamental physics of biomolecules and zoom out to the computational tools decoding them. Today, the field of computational biophysics is at a crossroads, and I'm exploring a major question. Where is the computational revolution actually stuck? So we've all seen the headlines. Deep learning tools are predicting static protein structures in mere seconds. Exascale supercomputers are pumping out nanoscale trajectories by the millions. From the outside, it looks like the computational biophysics game has been won. Game over. Biology solved. And thanks to a 7.5 million year calculation by the Deep Thought supercomputer, we even know the answer to the ultimate question of life, the universe, and everything. 42. Well, so long, and thanks for all the fish, Doug Adams, right? Anyway, that's fiction. But on a more real note, predicting a static snapshot of a folded protein is a world away from watching a living biological system actually breathe, react, fold, assemble, and translate in real time. So today, let's unpack the 10 greatest challenges in computational biophysics and biochemistry. The ultimate holy grails that keep structural biologists, computational chemists, and even software engineers up all night eating pizza, right? So let's break this down as a 3-act play, right? So Act 1 is the atomic frontier. That's where classical physics meets quantum reality. Act 2 is the algorithmic frontier. That's where conformational chaos and machine learning clash together. Act 3 is the cellular frontier. That's where we attempt the impossible, building a virtual cell. Actually doing a molecular dynamics simulation. Can we do that? That sounds complicated. So for each act, I've actually pulled a groundbreaking paper, fresh off the press, to show you how researchers are tackling these exact problems today. So grab a cup of coffee or your favorite fermented beverage, whichever your choice is. Fire up your favorite text editor, and let's get down to it. So let's start with Act 1, and that's the atomic frontier, and that's the physics problem. So we're starting at the smallest scale. That's the atoms themselves. Now, when you run a standard molecular dynamics, or MD simulation, you're usually relying on a classical force field. Think of all the atoms as tiny spherical billiard balls, right? And they're connected by spring-like bonds, so they can jiggle and wiggle, just like Richard Feynman suggested back in the 60s. It's fast, it's elegant, and for a lot of structural mechanics, it works surprisingly well. But biology doesn't play by simple classical rules, because remember, they're all made up of atoms, and the reactivity is where the electrons are. So that brings us to challenge number 1, and that's the multi-scale problem. Biological life happens across vast scales of space and time. A bond vibrates on a femtosecond timescale. That's 10 to the minus 15th, or 1 times 10 to the 15th, 1 over 10 to the 15th power of a second. The conformational changes in biomolecules and enzymes, or even cell signaling cascades, can take milliseconds, seconds, or even hours. How do you bridge femtosecond math with second scale biology without needing a massive supercomputer for all those calculations? Maybe a massive supercomputer the size of the moon. Which brings us to another question. Why aren't we building data centers in outer space? But that's a debate for a different discussion, right? Anyway, you have to seamlessly hand off calculations between quantum mechanics, atomistic MD, and coarse-grained models without actually losing critical physical details along the way. And speaking of quantum mechanics, that brings us right to challenge number 2, modeling quantum chemical reactions in actual biomolecules. Now, a classical force field, that's molecular mechanics, these force fields can't break or form chemical bonds. They don't know what to do with radical chemistry, charge transfer, metalloenzymes. If you want to watch an enzyme actually catalyze a reaction, you need a hybrid QM-MM method, and that stands for quantum mechanics / molecular mechanics. What you do is you use quantum mechanics for like the active site and molecular mechanics for everything else around it. And that's because solving the Schrodinger equation is computationally brutal, right? And biomolecules have thousands of atoms. And because the Schrodinger equation calculates where electrons are and what they're doing and how they're interacting, you've got 5 to 20 orbitals per atom that you have to calculate. In a protein, that's astronomical. Well, maybe Douglas Adams was right when he wrote the deep thought took 7.5 million years to answer the ultimate question of life, the universe, and everything. So that brings us to challenge number 3, and that's parameterization and polarizability. In most standard force fields, an atom's charge is fixed. Carbon is a carbon, and it can only form 4 bonds. Sorry, Texas. A nitrogen's a nitrogen, an oxygen's an oxygen, and so on and so forth up to 118 elements that we know of today, right? But in reality, electron clouds are kind of squishy. They're actually more like marshmallows instead of billiard balls. If you put a magnesium ion or a zinc ion near the active site, or if you put an RNA backbone in a dense salt environment, those electron clouds are going to shift. They're going to polarize, right? Fixed charge force fields fail hard in highly charged environments. And so moving the field toward polarizable force fields, such as amoeba, and amoeba is actually an acronym that stands for Atomic Multipole Optimized Energetics for Biomolecular Applications. Or you have neural network potentials. It's using artificial intelligence, neural networks. It's essential we ever want to, this is essential if we ever want to actually model true chemical accuracy, right? And that brings us to challenge number 4, and that's salvation, water entropy, and the overall cellular environment. Water is not just a passive background buffer. It's an active biological player. And quite recently, Andy Weir gave us a very interesting speculation through his character, Dr. Ryland Grace, portrayed by Ryan Gosling in Project Hail Mary. And they talked about the possibility of water not being required for life. It was rather devastating for Dr. Grace's career in the book. And what we kind of found, of course, this is just science fiction, but in the book, remember, Dr. Grace eventually found that astrophage and even rocky the iridium, while didn't appear to be water-based, actually involved quite a bit of water. And if you want to know more, you've got to read the book or go see the movie, right? Anyway, calculating the entropic cost of displacing water molecules from a binding pocket or modeling variable pH in real time is rather difficult. Water drives the hydrophobic effect, structural stabilization, and it's even involved in drug binding with bridging interactions and enzymes. If your water physics are wrong as well, your biological prediction is also going to be wrong. So if you want to see how labs are bridging this exact gap between quantum mechanical and classical speeds right now, let's take a look at a new publication by Valentin Gradistanu and colleagues. I think I pronounced the name right. I'm not sure. I apologize if I did, or if I didn't. But anyway, this paper is entitled Simulating Enzyme Catalysis with Electrostatically Embedded Machine Learning Potentials. If you want to read the actual potential, any paper I mentioned in this video, I will put in the video description. We can call those the show notes, and then you can link to that and you can read the actual papers. Now, the authors of this paper demonstrate that embedding machine learning potentials trained on ab initio quantum data into classical electrostatic environments allows us to simulate bond-breaking enzyme catalysis at near quantum mechanic accuracy. This skips the multi-week computational bottleneck of quantum mechanical calculations. And so this is actually an example of solving the atomic frontier in action. And that concludes Act 1, and that's going to bring us to Act 2 of our little 3-act play. And so that Act 2 is the algorithmic frontier, where we have conformations as well as coding, right? So now we've got our atomic force fields and our physics straight. So let's set up the middle scale, and that's conformational dynamics and data. Now, if AlphaFold actually taught us anything, it's that AI is incredible at recognizing static structural patterns. That's kind of what it's good at. It's not perfect. It certainly isn't great for a lot of things, but it does recognize patterns very well. But here's the challenge, and I've even talked about this in previous videos, right? Biology isn't static. Proteins are not statues, unless you stare at Medusa's snake-covered head for too long, or you're a weeping angel fighting the doctor. There's my Doctor Who reference. So proteins and biomolecules are dynamic ensembles of structures, right? It's a living, breathing construct. It's a living, breathing component. And so this is challenge number 5. And challenge number 5 is mapping conformational ensembles and rare transition states. A protein's function often lives in its high energy transition states or subtle cryptic pockets. These are conformations that only exist for a microsecond every few minutes. Standard molecular dynamics will spend about 99% of the time stuck at the bottom of a deep energy well, bouncing around the equilibrium state. Unlocking enhanced sampling, Markov state models, and generative diffusion architectures to map true thermodynamic ensembles is probably one of the most urgent goals in drug discovery today that needs to be solved if we're going to find new drugs and identify new proteins and new targets and that sort of thing. So what if globular proteins with neat energy wells were hard? What about the proteins that don't fold at all? These are challenges that need to be addressed. And that also brings us to challenge number 6, and that's intrinsically disordered proteins, or IDPs, and phage separation. A huge chunk of the eukaryotic proteome consists of IDPs, or disordered regions. They don't have a single 3D structure. They're very flexible. They exist as shifting, flexible clouds of conformations, often undergoing liquid-liquid phase separation to form biomolecular condensates inside the cell. Traditional force fields tend to overcompact IDPs. They treat them like broken globular proteins. And so designing computational methods that capture this dynamic disorder is a massive frontier in neurodegenerative disease research, a major challenge that we need to face. And even some of my own research right here at Tennessee Tech involving the ASK1 and the MAP kinase pathway with JNK3. We've got an intrinsically disordered region that's really challenging to model and really challenging to understand exactly how it's involved in binding. And that is potential for Alzheimer's and Parkinson's disease research. So now let's talk about RNA with challenge number 7. And so this involves predicting RNA dynamics and 3D tertiary folding. Now, protein structure prediction is at incredible milestones, but RNA is like the Wild West. RNA is hyperflexible. It's heavily self-repulsive due to the polyanionic nature of the phosphate backbone. And it's utterly dependent on specific divalent ions such as magnesium to fold into 3D shapes like riboswitches or long non-coding RNAs. Without deep RNA structural data sets, physics-based algorithms have to do the heavy lifting. We're still hunting for reliable, high-resolution models. Now, to see how generative AI is taking on these conformational nightmares, let's take a look at a paper by Palash Bara and Jagannath Mandal, first published as a preprint on bioRxiv, that's free, and it's now published in the Journal of Chemical Physics. And again, that paper, both of these papers will be linked in the show notes or the video description. Now, the title of this paper is Assessing Generative Diffusion Models for Enhanced Sampling of Folded and Disordered Protein States Across Scales and in All Atom Resolution, published in 2025. The researchers actually used denoising diffusion probabilistic methods, or DDPM for short. This is the exact same underlying AI architecture that's behind popular AI image generators such as DALL-E and Midjourney. And what they're doing is they're sampling rare transition states in disordered ensembles at all atom resolution. Instead of waiting for microsecond-long brute force MD simulations, the diffusion models let us jump straight to the hidden conformations of proteins such as alpha-synuclein. That's one of the proteins that's highly disordered in Alzheimer's disease and neurodegenerative diseases. So this is a huge advantage that we have to take into account, but it also comes with the same cautions and warnings about using AI in your models as well too. And so we're concluding Act 2 of our 3-act plan. So Act 3 is the cellular frontier, and that brings us to calculations of actual cells, like cell-size things. So far, a lot of these proteins, it's just a part of a cell. It's a tiny part. And the cell is very large. The cell is the basic unit of life, right? So now let's put this on to perspective. And this is where industry and academia meet So Act 3 is about application and scale. And so challenge number 8 involves quantitative free energy calculations for drug discovery. And so in pharma, the dream is pretty simple. You input a protein target, you feed in a library of small molecules, and you accurately calculate the binding free energy, or the delta G, the Gibbs free energy, to within 1 kcal per mole. This is what pharmaceutical scientists will refer to as the chemical accuracy. But free energy perturbation methods, we can abbreviate that FEP, it's still hampered by incomplete sampling. It's also hampered by slow side chain movements and even ambiguous water molecules. We talked about water before, right? And so achieving robust, fully automated free energy perturbation workflows across diverse chemical space is like the holy grail of rational drug design. So kind of one of the things we want to strive for and achieve, because that's going to help us design better drugs. And so challenge number 9 is using physics-informed AI and data integration. And so one thing we know about AI and machine learning is part of that. Machine learning is pretty fast, but it can also be kind of like a black box. And that black box can happily hallucinate physically impossible geometries. We've all seen AI hallucinations before. Meanwhile, physical simulations are quite rigorous, but computational very slow, right? The challenge right now is building physics-informed AI, neural networks that strictly respect conservation of energy, rotational invariance, and thermodynamics, while directly absorbing noisy experimental data from cryo-EM, NMR, and single molecule FRET experiments, and even more experimental data. And that ultimately brings us to the final mountain, right? That's our final mountaintop. We're like traversing Mount Everest here. And so that's challenge number 10. That's whole cell computational biophysics. So imagine an in silico cell, an environment where millions of proteins, nucleic acids, lipids, and metabolites crowd together, collide, and interact in real time. We're talking about scaling from exascale supercomputing to true whole organism simulation. And projects like simulating a complete viral particle or a minimal bacterial cell are underway. But doing this with atomistic or fine coarse-grained fidelity is the absolute frontier of computational science. And yes, we do have working proof of concept for this holy grail. And so our final Act 3 feature paper is led by S.J. Marink and colleagues at the University of Groningen in the Netherlands and the University of Illinois at Urbana-Champaign. And that's entitled Molecular Dynamics Simulation of an Entire Cell. This was published in Frontiers of Chemistry, and there's another paper published in Cell, at the journal Cell. So using a Martini coarse-grained force field, the team built the first full-scale molecular model of a minimal synthetic bacterial cell, which is JCVI Syn3A. And this is a synthetic bacterial cell that was engineered to contain the smallest set of genes, the minimal set of genes that's required to sustain independent life and self-replication. It's got only 473 genes across 531,000 base pairs. And it tracks millions of crowded biomolecules, lipid bilayers, genomic data, and ribosomes colliding inside a single simulation box. It's mind-blowing work. Now, I'd like to encourage you to check out the paper linked to in the video description. It's quite interesting where we're coming. Now, this is just a coarse-grained model, but I think all atom models are certainly going to be forthcoming, right? So what lies on the path ahead? From quantum mechanical bond breaking in an active site to simulating the crowded interior of a bacterium. We've got 10 challenges, and they prove that computational biophysics is not solved, and it's far from it. We've got a lot of work to do. We're living through one of the most exciting eras in history, where physics, computer science, biological data are all colliding to redefine what's possible. So again, check out the show notes in the video description for links to all 3 of the papers I mentioned. Actually, I guess it's all 4 of the papers. From machine learning potentials in chemical science to generative diffusion for intrinsically disordered proteins. And of course, finally, the Marink's whole cell molecular dynamics model of a simple synthetic bacterium. Got a lot of interesting papers to check out. So, which of these 10 challenges do you think we're going to solve first? Is AI going to crack RNA folding before we get polarizable force fields into routine production? Or are we going to finally be able to model an actual cell, even a human cell? So let me know your thoughts in the comments below. And of course, if you enjoyed this episode, make sure you hit the little like button down below and subscribe to my channel. Hit the little bell icon so that you're updated whenever I post new content. And please share this episode with a fellow researcher or student. I've got a lot of interesting content, and I've got a lot of other podcast episodes of Biochemistry Unfolded. So until next time, keep exploring the space between the atoms and the algorithms, and thank you for watching. You have been listening to Biochemistry Unfolded, from atoms to algorithms. Thank you for watching. We will see you again in the next video.